All right, now let's talk a little bit more about the context around MHC Class 1 and Class 2. So we're going to go through these sections. Let's see. And remember, the ones that are knocked out in blue are ones that we'll cover, and they'll help you put some context to these things, but they're not things we're going to test on directly. All right. So, all right, a couple vocabulary words, because why not? So, polygenic is when you're going to have several proteins of identical function. Whereas polymorphic is when you're going to have one gene and it's going to exist in different forms, essentially. So yeah, you can have lots of different versions of the same thing. For example, HLA A, B, and C, which are, I believe, all MHC class 1 molecules, each are very similar proteins. And so I would say these would be called a polygenic set. They're going to have the same function, which is presenting things. They're just going to exist in a slight variant, basically. Let's see. Or there are some cases where you actually have, for example, multiple tRNA synthases that act on the same actual gene. So that would be a more clear-cut polygenic. I guess the MHCs would be somewhere between polymorphic and polygenic, because you have multiple copies, but different variants of each copy. So there we go. So when they're throwing around those words, that's what it means. Okay, HLA is a designation for human MHC. And there are several types of these. They're going to be the A, B, and C versions of HLA. And so human leukocyte antigen just means MHC class 1 for the most part. And so you're going to have 6 of these things that you inherited from your parents. 6 of these maximum. There are some cases, I actually know one person where they managed to inherit the same copy from both parents. Yeah, just accidentally, nothing weird going on there, but it's a thing that can happen. Some of these are common enough that, yeah, it is certainly possible for that to happen. So when you see it written out, what you'll see is HLA, which means really MHC. A, so that's the type, A, B, or C. And you have 2 of each of those. And then it'll have a little star, and that's fine. The little star is always there. And then O2. So this is the main types. This would be called an A star O2 or A2 type. And then the O1 is a subtype on the end of that. And there would be multiple subtypes, but they would all be very similar to HLA-A star O2. That's the idea. Okay, so there are H2 genes. These are the MHC of the mouse, and yeah, they have their own designation for those. The human ones, I think, are going to be of more interest to you and to your future career, I am guessing, yeah. Although if you're going into laboratory science, yeah, you'll need to pick up the mouse equivalents. All right, next one are our IR genes, immune response genes. So it's an ancient term for genetic polymorphisms, like genetic differences in genes that control the amount of immune response. So nobody uses this anymore. Now that we know that it's actually different alleles of MHC that influence binding of peptides. So there we go. All right, interferon gamma, great. So I used to work with somebody who called this the master molecule. It is very heavily involved in a lot of different white blood cell responses. So it's going to be produced by helper CD4s, Th1 cells. It's going to be produced in large amounts by CD8 T cells and by natural killer cells, and is going to attract other cells of this type and have pretty broad functions. We're going to activate macrophages through this, for example, and it's going to act through a different interferon. There's a different interferon gamma receptor, rather than the interferon alpha receptor 1 and 2. Okay, the class 2 transactivators, so C, and it looks like II, but it's really 2TA. Roman numerals, yeah. So it's a protein that's going to activate transcription of MHC class 2 genes. Fantastic. And then class 2 deficiency is going to be a case where, yeah, genetically you are not able to present molecules on MHC class 2. Class 1 presentation would still work, so you would still have that side of the adaptive immune system functioning, but the class 2 side, which is going to drive, it's going to be involved with Th2 responses and with production of antibodies in particular, that is not going to function. And so people with this condition, there actually would be various ways to break the system and end up with this condition, but they're all going to have a very serious immunodeficiency disease. Okay, and non-classical MHC genes, we've already mentioned those. These are going to look like MHC genes in structure, but they are not going to be able to present peptides to conventional alpha-beta T cells. Okay, and so some of these are the ones that we covered earlier. They're called MHC class 1b genes, and they're going to bind to a smaller subset of molecules, but these molecules are going to be pretty widely expressed on bacteria or fungus, for example. Okay, so how do you end up with your MHC? Well, you inherit these, and so yeah, you've got your human leukocyte antigen A, B, and C. So these together are going to be your class 1. And these are going to, there are several studies now that show that there is a strong linkage between which type of HLA A, B, and C you happen to inherit, because there are many, many different variations of these. I think there are the most variations of HLA type B. I think there are several hundred, at least minor variants that are known. And it's smaller sets for A and C, if I remember correctly. But I think the book will probably have a chart coming up. Now, as far as your class 2, the guys that are going to present via MHC class 2, you're going to have all these things over here. So you're going to have your TAP binding protein, neat. And then these are your HLA, DM, DP, DPQ, and DR. And your actual TAP proteins are going to be located in here as well. So it's like the whole system that came in a genetic chunk. Probably through one sort of event. We got this transferred from probably some other kind of creature. Yeah, I am not sure exactly what. You get a similar chunk in mice, so this is probably a pan-mammalian adaptation, having this particular set of genes in this order. But you can see that we've gone a little step further. We've duplicated some of these genes a little bit further. And the mouse versions of class 1 are called H2D, L, and K, and usually it's a lowercase on those, I think. As opposed to HLA, A, B, and C, which just means MHC class 1, type A, B, and C. There we go. All right. And so, yeah, there are a bunch of proteins that are involved in the entire process. Basically, everything that we've been talking about is going to be classified together, not classified together, but located within the same vicinity, so that your MHC system is kind of genetically compartmentalized. Class 3 are actually interesting genes. These are not involved in class 1 or class 2 presentation. They don't actually present as far as I'm aware, but you have a bunch of genes in this area that are still very involved in the process of immunological presentation. So it's, it's related to class 1 and class 2, but in a different way. For example, the disease myasthenia gravis is believed to be associated with a mutation in this area. Okay, a little more vocab, fine. So polymorphism is, variability in a gene. So you've got this version, somebody else has that other version, and they're going to function slightly differently. So I guess polymorphism, as defined here, is when you've got so many different ones that generally you're going to have less than 1% chance of having any particular version of this. And that is the case with MHC class 1, particularly the HLA-B genes, I think are extremely variable. I think the A's and C's are also variable, but a little bit less so. So alleles are forms of the genes, like your variant versus my variant. Heterozygous are people that are going to have two different alleles. So when you happen to get a different one from your mother and father. When you get the same one from your mother and father, this can make it easier to find a tissue match, because these are the same genes that they would look for to determine if you're a bone marrow donor recipient pair, something like that, or a kidney donor recipient, something like that. Yeah, they're sequencing these MHC class 1 and particularly class 2 tends to be what they would look at. Let's see, your haplotype is which ones you have, and yeah, from one particular parent, and then you'll have two haplotypes that together make your full type, which will be heterozygous or homozygous at each particular allele. Codominant ones are when you have two alleles that are going to be expressed in roughly equal amounts. That's a good thing. And so that would mean if you got a different MHC class 1 type A from your mother and father, you would make equal amounts of both of those and have an equal chance of being able to present peptides on either one of those. That's useful in that it broadens the potential immune response quite a bit. Okay. Yeah, monomorphic is a gene that occurs only in one form as opposed to polymorphic. There are some genes where there will only be one possible constant region or one possible variant of a gene. We just don't have real genetic variation out there that still leads to a functional gene. I guess you could have broken and functional genes and still be monomorphic. All right, so human MHC genes, yeah, highly polymorphic. So the DP, DQ, and DR, look at all the different types there, MHC class 2. And so imagine if you had 1,200 different types of DRB out there, how are you going to find a tissue donor? I mean, you have to cycle through a lot of different people. Remember that everybody may have two different variants there, but at the same time, you may want to try to match both of those variants rather than just one. In general, the better hit leads to better organ retention, as far as I recall. Here we go. So A and C have lower numbers of variants. HLA type B of the MHC class 1 has by far the most, and yeah, thousands. Yeah, so that's pretty good. And then your class 1B genes, these are our non-canonical class 1 MHCs. And they exist in far fewer versions, but at the same time, yeah, these are out there and do contribute to immunological function. Okay, so I guess, in a way, maybe this all belongs at the start of the chapter rather than the end, but this is the order in which the authors put it, so we'll carry on in this order. Yeah, so you can inherit, different copies of the same gene and make different copies, which is great. You can have multiple genes that are essentially doing the same function, for which you only inherit one copy of them, but you'll have, one from your mom and your dad. It's just that they're basically the same gene that encodes the same protein. Or you can have a situation like we have with the MHC molecules, which is going to combine polymorphism and polygeny. So you have more than one copy, your A's, B's, and C's, your D, P, Q, and R, yeah, for example. And you're going to have multiple copies of each one of those. They can be slightly different. And so, yeah, with a bit of good luck and nice genetic mixing, you can end up with children that are fairly broadly protected because they have a fairly broad repertoire of things that they can recognize. Okay, so let's pause right here. Thank you very much.